Hello everyone! Today we are going to understand an important and fundamental topic in plant biology, seed and germination. Every plant begins its life as a seed, and germination marks the transition from a dormant structure to an actively growing seedling. Let us first understand what a seed actually is. A seed is a living structure that contains a miniature, undeveloped plant known as the embryo. The embryo consists of three main parts: the radicle, which develops into the root, the plumule, which forms the shoot system, and the Cotyledons, which serve as seed leaves and often store or absorb nutrients during early growth. The embryo is surrounded by a protective outer covering called the seed coat, which protects it from mechanical injury, desiccation, and infection. Inside the seed, there is stored food, usually present in the endosperm or in the cotyledons. This stored reserve provides nourishment to the embryo until the young plant becomes capable of photosynthesis and independent food production. Now let us understand the concept of dormancy. Dormancy is a resting stage during which the seed remains alive, but metabolically inactive. In a dry seed, the moisture content is typically around 10 to 13%. Due to this low water content, metabolic activities, such as respiration and enzyme action, remain minimal. This reduced metabolic state allows the seed to survive unfavorable environmental conditions, such as extreme temperatures or drought. Dormancy is therefore a survival mechanism. It ensures that germination does not occur until environmental conditions become favorable for seedling establishment. When the seed receives adequate water, oxygen, and a suitable temperature, dormancy is broken and germination begins. Now let us define germination precisely. Germination is the sum of all physiological and biochemical processes that begin with water uptake by the seed and culminate in the emergence of the radicle through the seed coat. The appearance of the radicle is considered the completion of germination. The first stage of germination is imbibition. Imbibition refers to the absorption of water by the dry seed. Water enters the seed due to differences in water potential, moving from the surrounding moist soil into the seed tissues. As water is absorbed, the seed swells and rehydrates. This is primarily a physical process. It is important to note that even non-viable seeds can undergo imbibition because it does not initially require metabolic activity. Water uptake during germination follows a characteristic three-phase pattern. In phase 1, there is rapid water absorption. This corresponds to the initial swelling of the seed. In phase 2, water uptake slows down and reaches a plateau. During this phase, major metabolic processes are activated. Enzymes become functional, cellular repair mechanisms begin, and respiration increases. In phase 3, water uptake increases again, coinciding with radical protrusion and active cell expansion. Parallel to water uptake, there is a change in respiration rate. During phase 1, oxygen consumption increases sharply as metabolism resumes. In phase 2, respiration stabilizes temporarily. In phase 3, respiration increases again due to active cell division and elongation. Once leaves develop and photosynthesis begins, respiration gradually stabilizes as the seedling transitions toward autotrophic growth. Following imbibition, we enter stage 2, activation of enzyme systems. During this stage, pre-existing enzymes become active, and mitochondria resume efficient ATP production. Cellular structures that were partially damaged during desiccation undergo repair. Although enzyme activation occurs here, large-scale mobilization of stored food is still limited. Next is stage 3, mobilization of stored reserves. In this stage, new enzymes are synthesized, either from stored messenger, RNA, or newly transcribed, RNA. These enzymes break down stored macromolecules. Amylases convert starch into simple sugars. Proteases break proteins into amino acids. Lipases hydrolyze lipids into fatty acids and glycerol. Phosphatases release phosphate groups necessary for nucleic acid and ATP synthesis. these simpler molecules are transported to the growing embryonic axis, especially to the radicle, supplying both energy and structural components required for growth. Then comes stage 4, radical emergence and seedling growth. The radicle is the first structure to emerge from the seed coat. Its emergence marks the completion of germination. After this, active cell division and cell elongation occur in both root and shoot regions. The radicle grows downward into the soil, establishing anchorage and absorbing water, while the plumule grows upward to form the shoot system. At this stage, the seedling structure becomes more defined. The portion of the embryonic axis below the cotyledons is called the hypocotyl, and the portion above the cotyledons is known as the epicotyl. These regions contribute to proper shoot development. As the leaves expand and begin photosynthesis, the seedling becomes autotrophic. It gradually stops depending on stored reserves and begins synthesizing its own food using sunlight, carbon dioxide, and water. Finally, if we look at the overall process as a cycle, it begins with a dry, dormant seed, progresses through water uptake, enzyme activation, food mobilization, and radical emergence, and results in an independent seedling. This seedling eventually matures into a plant that produces seeds again, completing the life cycle. In summary, germination is a highly organized, energy-dependent, and sequential biological process that transforms a dormant seed into a self-sustaining green plant. So today, we discuss the entire process of seed germination in a clear and systematic way. From dormancy and imbibition to enzyme activation, reserve mobilization, and radical emergence. Remember, germination is not a single event, but a series of coordinated physiological and biochemical changes that begin with water uptake and end with the emergence of the radicle. If you found this explanation helpful, stay tuned to the channel for more clear and concept-based lectures. If you have any questions, feel free to mention it in the comments section. Thank you for watching and keep learning.